This is a production of Cornell University.
So thank you so much for the introduction, and thank you everyone for joining this horticulture section seminar. I'll be talking today mostly about soil and not so much about plants. I will have a small section about root exudates toward the ends of the presentation. So right to it. I'll speak shortly about my PhD work, in which I studied basically organo-mineral interactions, but in a polymer clay type of system. Then I'll discuss a couple of projects that I've been doing in the Lehman lab, looking at soil abiotic conditions and how they affect carbon cycling, mostly focusing on soil water content and some aspects of mineralogy. And then I'll discuss some experimental plans and setups that we have for studying the role of root exudates in the rhizosphere model. So, as Don said, I'm a soil scientist, but strangely, maybe my PhD focused not so much on soil science, but on functionalizing clay minerals to be used as sorbents to remove contaminants from water. So our working hypothesis was that if we take smectite, our typical 2-to-1 clay mineral, which is already a high surface area particle and is a pretty good sorbent for inorganic cations, and we functionalize it with different polymers that have different properties and chemistries, we can then achieve really great removal of a slew of organic contaminants that are detected in wastewater and different surface waters, and also that we can include some cool designs and functions on this sorbent, such as catalyzing the destruction of adsorbed pollutants on the surface and releasing them as less harmless byproducts of these harmful contaminants, such as organophosphate, pesticides, and others. And so just a short example of how what this looked like. In one of these projects, we showed that if we change the chemistry of the solution of the polymer, we can induce changes in the way the clay mineral and the polymer chains interact. So basically organo-mineral interactions, but on a clay polymer system. And so we transitioned from, let me go to laser. So we went from this flat polymer configuration on the clay, which we call the trains configuration, into a more extended loopy configuration that we call a loops and tails type of configuration. And of course, this means that there are more monomers dangling into solution, and that had implications for how this material was absorbing and interacting with other organic pollutants in the solution. So this was my last year of my PhD. I was thinking about doing a postdoc, but at that time I was mostly reading papers in the field of material engineering and research and water treatment engineering, etc. But I was still in the soil and water department when this article, basically by mistake, landed on my desk. This hugely cited 2015 paper by Johannes Lehmann from the Soil and Crop Section and Marcus Kleber from Oregon State University. And in this paper, and so just to say that I was pretty much out of touch from the soil science literature at that time, but in this paper, they synthesized recent data that called for a change in the paradigm of how we think of the soil organic matter. And so in the more conventional, older approaches, the focus was on substrate chemistry. That is, the decomposition of soil organic matter was seen as mostly a function of the chemistry of the organic matter. So different hypotheses were put forward and tested, such as the humidification hypothesis, selective preservation of recalcitrant materials, progressive decomposition, etc. But these hypotheses were somehow missing the fact that all of this was happening in a soil matrix. Whereas in this paper, the framework that was put forward and suggested was that organic inputs from plants and animals were continuously being processed by decomposer communities into smaller and smaller compounds, which are becoming more and more oxidized. And as these compounds were becoming more smaller and more oxidized, their affinity to interact with mineral surfaces or to go into soil aggregates increased. And so this organo-mineral interactions in aggregates or on mineral surfaces decreased their availability to the decomposer community. And all of these arrows are a function of different abiotic and also biotic parameters such as moisture, temperature, biology of the soil, uh, microbial community, and others. And so this framework was much more holistic, taking into account all of these different processes uh, that go into soil organic matter dynamics. And I thought, wow, how cool is this? This is where I want to be, especially because organo-mineral interactions were such a large part of this. And I said, let's go straight to the source, and was fortunate enough to land a postdoc scholarship and work with Johannes Niemann on this subject. So another fundamental understanding of this new framework was that microbes in the soil not only are agents of decomposition of organic inputs in the soil, but uh, they also constitute many times the majority of soil organic matter. So, for example, in this important paper from 2016, we see the distribution of organic compounds basically in different classes on a clay mineral that was added, that had glucose added to it, and then was incubated with a microbial inoculum for 18 months, and then was analyzed again. So this is basically a clay mineral, sugar, microbes, and the organic composition is now strikingly resembling that of a field soil, which indicates that soil organic matter must be mostly made out of organic of microbes, their necromass, and the microbial metabolites that they exude throughout their lifetime. A visual piece of evidence for this is from an image taken by Johannes using soft X-ray spectral microscopy technique called sticks and MXFs. And here we see a microbial cell in dark green, surrounded by what is probably microbial metabolites. And it's interesting to see that these microbial metabolites coat the different soil, mineral, and black carbon particles in the soil. So this just illustrates how soil organic matter is the product of microbial decomposition of organic inputs and the microbial themselves. And this coating of microbial metabolites on soil particles is what we understand is the basis of organo-mineral interactions, which protect soil organic matter from further decomposition and enable some of these compounds to persist in soil for decades and centuries and sometimes even more. So after this very short intro, I'll show you some of the projects that I've been working on, focusing on soil abiotic conditions and how they affect carbon cycling. So looking at water, we know what the immediate effects are of water content on decomposition of soil organic carbon. So when we increase the amount of water in the soil, there's an increased water activity and a greater rate of diffusion of nutrients and substrates to the decomposer community. And this is reflected in higher sea loss here as respiration and CO2. But when we increase the water content even further, oxygen diffusion now becomes a, a, uh, a limiting factor, sorry, which is reflected in a decrease in carbon loss. But although the immediate effect of water content is known, the long-term effect of water content is less known. And so to try to probe this phenomenon, we looked at an experimental plot uh, close to campus on Hanshaw Road. And this plot has been used and tested for different bioenergy crops. As part of this campaign, the soil water content has been uh, getting a chat here, but I'll ignore it for now. Has been measured many times, and it's been established that there is a moisture gradient across this field. So in the central part of the field, these blue dots, this area is relatively wetter than the outer regions of this field, which are relatively drier. So for each subplot was assigned sort of a normalized moisture value dependent on a multi-annual relative moisture calculation. And so one of the observations from previous work was that with increasing moisture values in the soil, there was an increase in soil organic carbon and also of total nitrogen. And so we thought this is a unique location to understand how long-term moisture gradient affects this accumulation of carbon and try to understand the mechanisms underlying this accumulation of carbon. So we hypothesized that there could be several different processes working basically at the same time, which could affect what we're seeing. So with increasing soil moisture, there could be increase in the reductive dissolution of mineral phases that are redox sensitive. When there is more water, there's greater opportunity for element transport, vertical and lateral. With increased soil moisture up to a certain point, there is an increase in microbial activity, as we just saw. Soil organic carbon becomes more oxidized due to this decomposition of microbes. And then as organic compounds become more oxidized, they generally have higher affinity to absorb on soil minerals because they become more charged and more likely to interact with minerals. So these changes, which we hypothesized are working across the mineral and the organic components of this organomineral interaction idea, we thought was probably what was causing the differences in soil organic carbon content across the moisture gradients. So we sampled a bunch of soils in this experimental plot and measured several things, tried to understand this. We measured the microbial biomass. We did a density fractionation on the soil, which allows us to isolate the particulate organic matter, POM, which is the fraction that is most resembling plant litter. It's the least decomposed pool. So the free POM is the POM that is not within aggregates. The occluded POM is the POM that is occluded inside aggregates. And we can also isolate the silt and clay heavy mineral fraction and the mineral associated organic matter, MAOM, associated with that fraction. We extracted soil oxides and hydro oxides with a sequential extraction scheme, and we measured the concentrations of aluminum, iron, and carbon that are stored inside each of these oxide pools. And we also performed soil incubations to measure how much CO2 is being inspired from these different soils and whether they relate to different soil properties. So today I'll discuss a little bit about the microbial biomass, the silt and clay fraction, the oxide, the soil oxide pool, and some of the results of the soil incubations. So first of all, we did a sequential oxide extraction in which we extracted increasingly crystalline phases of the soil, and I'll walk you through these results. So as a function of normalized moisture values, we have the extractable aluminum, extractable iron, and extractable carbon concentrations in the different extracts. We don't have to think now about what color is which. The aluminum concentration was basically constant across the different normalized moisture values, and this is because aluminum phases are usually not moisture sensitive and not redox sensitive. Iron phases usually are, or some of them are, and we saw that the more non-crystalline amorphous phases decreased in their concentration in the wetter soils, and the crystalline phases, the more ordered minerals, increased with increased moisture. And this has been reported in the literature, and the explanation is that wetter soils experience more events of reducing conditions in which iron is reduced, dissolved into solution, and then when soils re-dry again, some of that iron recrystallizes into a more crystalline and orderly type of phase. So this is explained by that. And then the carbon concentration across moisture largely follow the patterns of iron speciation. So with the wetter soils had more carbon released from the crystalline phases and less carbon released from the non-crystalline phases. So you see that moisture had a pretty substantial effect on iron speciation and how much carbon was being stored in each type of phase correspondingly. And then we plotted the carbon um, against the iron and against the aluminum extracted from each one of the extractions to try to understand which of these elements was more important. And so the non-significant regressions with iron, but the significant regressions with the amorphous aluminum phases show us that it was the aluminum um, amorphous and organometal complexes formed between carbon and aluminum were the ones that were driving storage of carbon in these soil oxide pools. And this also has been increasingly been shown in recent literature looking at how moisture is affecting oxide interactions with carbon. And then we are now looking at some aspects of microbial processing and cycling of the soil organic carbon. So this is the carbon to nitrogen ratio of the mineral associated organic matter fraction. So the carbon to nitrogen ratio decreases with increasing moisture, and then the microbial biomass normalized to soil carbon increased with increasing moisture, and the dissolved organic carbon, just the dissolved carbon substrate in the solution, that also decreased with increasing moisture. So how can this be explained? So higher moisture supported a higher microbial biomass and more microbial activity in the wetter soils. This probably led to perhaps more uptake of dissolved organic substrates, and the increased microbial processing of these substrates by the larger microbial biomass led to the deposition of organic matter on the silt and clay fraction that was more highly processed and more microbial in signature, and therefore had a lower carbon to nitrogen ratio. So we see that wetter soils had more microbes, more microbial activity, and a mineral associated organic matter fraction that was more microbially processed. And then we did FTIR spectroscopy to look at how these changes in processing of carbon reflected on the chemistry of the carbon. And so we have the three spectra are averages of soils from high, mid, and low moisture levels. And when the absorbance of a peak is higher, that means there is more of that functional group in this specific wave number in the soil. So we'll go from left to right. In the wetter soils, we had more OH, so more phenolic groups and more perhaps carbohydrate groups. We had more aliphatic carbon, and we had more carboxylate and aromatic carbon, and also a relative increase in aromatic carbon to nitrogen, carbon-nitrogen bonds, and NH bonds. And most of these are usually assigned to organic matter that is more microbial, more processed in nature. And while FTIR is a great sort of exploratory type of analysis that allows us to understand maybe differences that are happening in composition and amounts, it is difficult to obtain quantitative numbers from this type of analytical technique. So we turn to carbon NMR on the right and carbon NEXAPS, which is another type of spectroscopy that we like to use for carbon. And we looked at the rate, the ratio of carboxylic carbon to aromatic carbon in the MAOM fraction, the silt and clay fraction of soils from low, mid, and high moisture levels. And so this ratio, again, reflects the extent of usually the decomposition of plant matter into microbial carbon. And so we see that with increasing moisture, the ratio of carboxylic carbon to aromatic carbon increased, and that is shown with carbon NMR and carbon NEXAPS, which means that in wetter soils we had more decomposition, and the organic matter was more microbial in nature and less plant-like, and that corresponded with decreasing carbon to nitrogen ratio. So all in all, wetter soils had more microbes, more processing of organic carbon, and that carbon was more oxidized. And then to try to relate the different variables that we measured in the soil, some of these I discussed and some I didn't, with three important parameters. One of them is soil organic carbon, the contents of the carbon, mineralization, which is the result of what we find in an incubation, the amount of CO2 that's released from the soil, and mineralizability, which is that CO2 normalized per unit carbon, which reflects the persistence of that soil organic carbon, its resistance to decomposition. So we have a correlation heat map here, and also to try to find groups or sets of variables that best explain these three parameters. We constructed several partial least square regression models, and these models are great for dealing with data where we have less observations of variables and variables that are very multicollinear. So I've highlighted only the predictors that had significant coefficients in the model. So soil organic carbon stocks were best explained by mineral protection in exchangeable calcium and amorphous aluminum phases, and by the amount of litter as FPON in the soil. Soil organic carbon mineralization is basically a function of carbon and litter, which is what we usually expect. And soil organic carbon persistence was linked to a combination of microbial residues, here portrayed as the carbon to nitrogen ratio, and protection by reactive iron and aluminum phases, here the aluminum pyrophosphate and iron pyrophosphate. So to conclude all of this first project, we saw that the capacity of the soil to accumulate and store carbon was a combination of uh, microbial decomposition of plant input and the soil mineral phase capacity to absorb and store these microbial byproducts. So moving on to the next project, here we specifically wanted to look at the role of calcium in this story. And this has been a focus in the community for some recent years. Here, in a biogeochemistry review from 2018 by Mike Rowley, shown that as soil pH increases, the relative importance of aluminum and iron phases in storing and stabilizing carbon decreases, whereas that of calcium increases. And this is just because of how soil chemistry uh, is transitioned across pHs. And so these interactions with calcium can be in the form of direct sorption to calcium carbonate, interactions with calcium ions that are themselves absorbed on clays, aggregation and flocculation, and physical protection that is mediated by calcium and others. And so in soils that are neutral or slightly alkaline, in which calcium plays an important role, we often see that with the more calcium we have, the more organic matter content that we have. And so we wanted to further explore this relationship and try to find specifically some mechanisms for this. Um, and so other studies have also shown that other mechanisms may exist. So for example, in this incubation of a organic horizon in which soil minerals are largely absent, the addition of calcium resulted in decreased, here in black, in decreased mineralization of the organic carbon. So because there are no minerals here, organo-mineral interactions probably play a very small part of what is going on here. So perhaps there are some organo-organic interactions in which calcium is playing a role. And here, this is data from Dan Buckley's lab, unpublished, I think, shows that addition of calcium here in the form of addition of liming drives a shift in the microbial community in soil. And that shift is also associated with a decrease in the activity of some degradative enzymes and respiration. So we see that overall calcium may have very different roles and additional roles in addition to just organomineral interactions. And this is what we're very much interested in investigating. Quick sip of water. And so the experimental setup was the following. Well, we took a soil and we pre-incubated it with calcium chloride that was enriched with the 44 isotope. Um, and during this pre-incubation, we caused a 10% increase in exchangeable calcium. So some of this labeled calcium went into the mineral matrix and kicked off some other cations. And then we leached the soil of excess salts. And of course, we did this with control soil, but without calcium, just with water. We air dried the soils, and then we added 13C15N leaf litter from willows, and we incubated this litter soil mixture at 40 and 60% of their water holding capacity to continue our um, interest in looking at how water content is affecting all of this on top of the role of calcium. And this incubation was carried until 50% of the plant litter was mineralized approximately. And throughout the incubation, we measured CO2. We also have some delta 13C CO2 measurements. We sampled the incubation at the beginning and end of the incubation, and we isolated different fractions that I also showed in the previous project and measured carbon and nitrogen elemental percentages and isotope ratios to try to follow this labeled litter tracer into the different pools. And we also sent out samples for carbon and calcium nexaphs. We did FTIR microscopy to try to get some spatial distribution of minerals and organic matter across these samples. And we are also hopeful to get some nanosims images in the summer. So nanosims is basically a type of scanning electron microscope that can also identify and measure the relative abundance of different elements. So we can hopefully uh, locate where the labeled calcium is in relation to labeled carbon and nitrogen, native carbon and nitrogen and other soil minerals. And based on this spatial distribution of these elements, try to understand further the mechanisms underlying what calcium is doing in soil, which is what I'll be showing now. So this is the cumulative mineralization in CO2 carbon per gram soil across the incubation. So we have the low water content treatment, the high water content treatment. We have the soils without litter and with litter. And for each one of these curves, we have a control, DW, and a calcium treated soil. What we can see first is in all of these combination, except for the no litter high water content, calcium caused a decrease in the CO2 emitted. So some sort of stabilization was occurring when we added fats into the soils. At the beginning of the incubation, at day 4, so I forgot to say, we destructively sampled at day 4 and at day 130. At day 4, around 80% of the CO2 originated from litter carbon. So the microbes were busy hacking away at the litter, decomposing it, and some of that CO2 was being released. And this was um, similar across the different experimental conditions. Towards the end of the incubation, less of that CO2 was from the litter and more of it was from the soil organic carbon, but there was also a difference between different treatments. So in the low water content treatment, now only 65% of the CO2 was originating from the litter, whereas in the high water content treatment, 70 to 75% of the CO2 originated from the litter. So we can say here that the high water content treatment um, favored the utilization of litter as a substrate relative to the low water content. And looking into further detail, we can also look at the amount of POM in the free and occluded pools with the different water contents. So we can see that the high water content treatment increased FPOM decomposition because of this difference in the end time point of the FPOM content, whereas low water content increased the decomposition of OPOM. Here again, this difference between these two box plots signifying the amount of OPOM at the end of the experiment. And so You can see that water content not only drove litter decomposition, but it also affected where that litter in the soil was being decomposed. And to try to explain that, we can look at this scheme of the soil matrix, and let's imagine for a second that this is a free PON particle that is in the interaggregate space, not confined by clay minerals. Uh, the pore space, the pore size diameter in these spaces is usually quite large, and under a low water content treatment, the water here will mostly be drained away, and there will likely not be enough water to enable good decomposition of FPOM, which is why its decomposition is favored at high water content. Whereas if we imagine this is an occluded POM particle that's confined within these clay minerals, the pore size diameter of these locations is usually much smaller, and they can retain water even at relatively lower water content, which explains why at low water content, this type of POM must be targeted preferentially. Looking now at the cycling of litter into the microbial biomass, we find some interesting things. So under calcium treatment at the beginning incubation, we find that the fraction of microbial biomass carbon from litter increased relative to control soils. And this was highly evident at the beginning of the incubation and slightly less so towards the end of the incubation, but still significant. So we added calcium, and more litter was being incorporated into microbial biomass carbon, cycling into microbial biomass carbon. And this was also associated with higher carbon use efficiency under calcium treatment. So we added calcium, and per unit carbon of litter, more carbon was being assimilated and turned into microbial biomass than CO2, then carbon lost as CO2 in the calcium treatment. Again, very evident at the beginning of the experiment, and somewhat less so towards the end of the experiment. So we see here that calcium favored the efficient incorporation uh, of the litter carbon into microbial biomass carbon, and we know that this is the pathway towards stabilized carbon. And now looking at the litter carbon that has found its way after being processed into the silt and clay fraction, we thought that calcium would drive this upwards, but counterintuitively, we found the opposite. So calcium treatment caused a lower percentage of carbon in the MAOM pool to originate from the label relative to the control DW soils at the beginning of the incubation. At the end of the incubation, this sort of evened out, and in the low water content, we still saw this difference. So at the beginning, less carbon from the label reached the certain clay fraction, probably because more of it was still being cycled through the microbial biomass carbon. And then looking at the C to N ratio of the label on the silt and clay fraction. So at the beginning of the experiment, there was not a lot of differences or mixed differences across the water content treatments. But then at the end of the incubation, after a lot of this processing has already occurred, we can see that the C to N ratio of the soils treated with calcium was significantly lower than in the soils not treated with calcium. So this shows that although carbon from the label did not immediately reach the silt and clay, it first uh, stopped, I guess, at the microbial biomass. When it was finally deposited on silt and clay particles, it was more highly processed, perhaps containing microbial necromass and microbial metabolites with lower C to N ratios. Those were being deposited on the silt and clay fractions in soils treated with calcium. So to try to put all of these different findings together into a mechanism-like scheme, uh, we saw that plant litter was being incorporated into the FPOM or the OPOM pools. The relative contribution of these is likely controlled by aggregation and disaggregation, though this is not something that we looked at this specific project. We saw the high moisture uh, favor the decomposition of DPOM, while low moisture favored the decomposition of depleted POM. Soils treated with calcium uh, had a higher incorporation of decomposed litter into the microbial biomass and preferentially more deposition of highly processed microbial necromass and probably extracellular metabolites on the silted clay or MAO fraction, uh, whereas the control soils uh, had relatively more deposition of partly decomposed litter um, on the silted clay fraction. And putting everything together, this resulted in higher carbon to nitrogen ratio on the MAOM, lower carbon use efficiency, more CO2 emitted in the control soils, and lower carbon to nitrogen ratio, higher carbon use efficiency, and less CO2 emitted when we treated the soils with a calcium salt. So moving forward, and I hope that I'm pretty much on time, I'll share some experimental plans that we have for studying the role of root exudates in rhizosphere carbon. Unfortunately, I don't have a lot of results, but I do have some uh, cool image and some interesting plans. So several studies have tried to quantify root exudation, and it is said that anywhere between 3 and 11% of the carbon that plants fix are allocated or lost, depends on how you look at it, as root exudates. So a substantial amount of carbon entering the soil is in the form of root exudates. At the same time, we also think that the injection of soluble root exudate compounds into the rhizosphere, which is highly populated by microbes, is has a higher chance of resulting in carbon that is microbially processed and therefore can persist long term in the soil relative to above ground plant litter that is sometimes deposited on areas that do not necessarily have high microbial population. So this point of entry and solubility of oxides make this carbon pool, root exudates, very interesting in the point of view of a soil scientist looking at the soil organic carbon uh, dynamics. Another thing is composition. So this is metabolite profiling of a rhizosphere soil and a bulk soil in a maize field. So you see that um, because of exudation and other root processes, the composition of the soil solution is vastly different in the rhizosphere soil versus the bulk soil. So this combination of different chemical composition, the solubility of the compounds versus solid compounds, the high density of decomposers, and intimate interaction with soil minerals make the rhizosphere sort of the most interesting place to be right now for studying soil organic matter. And thinking again about soil abiotic conditions and specifically water content, it's been shown that drought can cause a change in the composition of root exudates. So here, in a normal treatment, 80% of exudates were primary metabolites. And here 70% of the exudates were secondary metabolites when these plants were subjected to the drought treatment. In another paper shown here, drought-induced exudation, where these exudates were then collected and put into another soil that did not experience this drought, that resulted in increased soil organic carbon mineralization. So you see that again, abiotic conditions can result indirectly um, in differences in soil organic carbon cycling. There are many unknowns across this interface of plant roots, soil, and carbon. So, for example, while in the previous project we showed how soil abiotic conditions can regulate litter turnover, this information is largely missing if we're talking about turnover and cycling of food exudates. We don't really understand the mechanism underlying higher mineralization with drought-induced exudation. We would like to know more about which compounds or compound classes are involved in drought-induced mineralization. All of these questions are embedded in the research plans that we have ahead. So to try to understand this role of roots and root exudates in carbon cycling, we've um, thought of several methods and some experimental setups. So the first is this microcosm, which we have designed, in which the CO2 respired from the soil is trapped within this headspace, and that headspace is isolated from the rest of the atmosphere so that we can measure the soil CO2 respiration without it being mixed with the outer atmosphere. So, for example, if we take a C3 soil, or a soil that has mostly C3 vegetation, and grow C4 plants such as maize, so using a isotopic mixing model, we can calculate the amount of exudates, and we can partition those exudates as they go into CO2 and different soil and microbial pools, and try to understand their turnover under different experimental conditions. And then to try to probe the composition of the rhizosphere solution as roots grow and compare it with bulk soil, we are going to use microdialysis probes, which are basically these tiny tubes that have a semi-permeable membrane, and we pump a solution through this tubing, usually just water or some salt solution, and as the solution flows, the solution in the soil diffuses, or some of these solutes diffuse into the membrane, then we can in situ probe and collect the solution with minimal interference. And then to try to locate the probe in relation to the roots, we can use micro CT. So this is an image taken here at the Biotech Institute, the imaging facilities. So this is a soil core, like you saw before, that acrylic tube. And here we can see that the tubing and the microdialysis probe inserted into the soil. These white specks are quartz grains inside the soil. And we are also now looking at how we can identify the roots. So we can, as a function of the density of the roots or the proximity of the roots to the microdialysis probe, we can now try to relate the composition of compounds in the rhizosphere to the proximity of the roots. So those are the questions that are now being asked by us. And so with that, I will finish up. Thank you so much for giving me your time and listening. I'd like to thank the different research groups that I have had the opportunity to work with, different analytical and service labs, and some funding agencies. And with that, I'm very happy to take any questions that you may have. Thank you so much.
Let me ask if there are questions from the group for Itamar. And if you have a question, just unmute yourself. I am not seeing any questions, so Itamar, I will ask one, which is in your experimental work, either previously with Johannes Lehmann or currently with Taryn Bowerly, have you looked across a spectrum of soil types, clays, loams, sandy soils, to determine how the organic fraction behaves?
Yeah, so in another project that I did not discuss at all here, we have a collection which we call the National Soil Collection, mostly collected by Rachel Acqua, a PhD student in Johannes Lehmann's group, where we have soils from 70 or 80 different locations in the United States. So we are exploring many different relationships between soil organic carbon pools and cycling, and trying to relate those to many different variables that we see in the soil. So yes, the short answer is yes, but did not show
Thank
it
you.
here.
Other questions from the group? let's see, I'm not seeing any, so I want to thank you, Itamar, for that very well done seminar. And along with that, I want to wish everyone a happy end of the semester and summer, in that this wraps up our seminars for spring 21, as well as, I should mention, my 8 years of running this seminar. So thank you all. Bye-bye now.
Thank you. Bye-bye.
This has been a production of Cornell University, on the web at cornell.edu.